the Mind and Matter podcast. I'm your host, Nick Jikomis, and today I'm speaking with Dr. John Cryan. John is a professor and chair of the Anatomy and Neuroscience department at the University College-Cork in Ireland. His lab studies the microbiota-gut-brain axis in health and disease, and we touched on a range of topics related to how the gut microbiome affects the brain and body, including how the microbiome affects aging and immunity, such as what happens when you actually transplant the microbiome from a young individual into an older one, how the microbiome affects social behavior, including potential links to autism, how antibiotics and the general hyper-cleanliness of the modern world impacts our microbiomes, and things like how the microbiome affects neuropsychiatric conditions like depression, as well as how diet influences brain inflammation. Towards the end, John also shared an interesting example of a food that he consumes for gut health, and we touched on a variety of topics in this general space of the gut microbiome and how it connects to the brain and general health. As always, if you enjoy the content on this podcast, please like, share, and subscribe wherever you're listening or watching, if you're watching the video version on YouTube. There's also a variety of links in the episode description that go to places like my Substack, mindandmatter.substack.com, where you can find both the podcast and some of my long-form writing related to topics that I cover on the show. You'll also find links to a few different products and services that I use and recommend. And if you have a minute, I encourage you to check those out. They're pretty interesting, and anything that you purchase through those links will directly support the podcast and help keep it going. Hey everyone, I want to take just a minute or two to discuss some interesting new tech you can use for blood glucose, that is blood sugar monitoring. I've discussed blood glucose and the physiological effects of diet on the podcast many times, and I want to share my experience using Levels Health to monitor my blood sugar levels with an easy-to-use app. People with diabetes have to regularly check their blood sugar levels to make sure they stay within a healthy range, but glucose tracking can also be valuable as a tool for non-diabetics and people in good general health. For example, I've used this Levels product on a couple different occasions to understand how different parts of my own diet were impacting my blood sugar levels. I learned that certain foods I was consuming were causing large blood sugar spikes, even in cases where the food didn't contain that much added sugar, and I never would have guessed it. So it actually enabled me to cut certain things from my diet that I wouldn't have otherwise cut out. Different foods cause very different patterns of blood sugar change, at least for me, and I had no idea about this before I actually started tracking it. Levels would also be perfect for anyone who wants to experiment with changing their diet, because it would allow you to actually see how dietary changes are impacting your blood glucose levels. It's a blood glucose monitor that you affix to your upper arm. It's really easy and painless, and the Levels app allows you to take readings of your blood sugar throughout the day. One monitoring device lasts for 2 weeks, which is more than enough time to understand your own physiology with precision. Levels is currently running a closed beta program with a wait list of over 160,000 people, but you can skip to the line and join Levels today in this beta program by going to the link levels.link/mindandmatter, spelled out all one word, which you can also find in the episode description. Even if this is a product you use just once or twice or intermittently as you change your diet, it can provide you with valuable insights into how your diet is impacting your blood glucose levels. So again, check out the link to the Levels product in the episode description. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Today's show is brought to you in part by Dosist. an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. And with that, here's my conversation with Professor John Cryan. Professor John Cryan, how are you?
I'm good, Nick.
Can you tell everyone where you're calling in from and what you do scientifically?
Yeah, so I'm calling in today from Cork, which is the second city in Ireland, right at the very south of Ireland. And I'm a neuroscientist, of course, but I am intrigued by how the microbiome in our gut influences our brain and behavior.
Interesting. And so is that basically what's referred to by this term microbiome-gut-brain axis?
Exactly. That's, this axis is now becoming part of the mainstream aspects of physiology now. We always had a gut-brain axis, and it's in the last 2 decades that there's become a greater appreciation that there's another component to it, and that the microbes are actually key to how the gut-brain axis works.
What is the gut-brain axis? What is it? What are the physical connections mediating how the gut and the brain communicate?
Yeah. So, I mean, this is, we know a lot about this from basic, physiology going back to Pavlov's time, about understanding about how our gut is talking to our brain. And we use it in our language, the gut feelings aspects of it. The main pathways of communication between our gut and our brain are neural. So that will involve through for example, the vagus nerve. Vagus comes from the same derivation as words like vagrant and vagabond. So it means wandering. Mm. And so this is a long wandering nerve, one of the cranial nerves. And vagal signals go in 2 directions, to the brain and from the brain to all the organs, including the gut. We also have spinal mechanisms that are at play. And within the gut, from a neural perspective, we have what's called the enteric nervous system, the ENS, and this is the second brain, as Mike Gershon at Columbia once wrote a book about, and we have more nerves in this second brain than we do in our spinal cord, and so this is this enteric nervous system plays a key role in how the gut functions from a basic digestion physiology aspect, but also more recently we're beginning to appreciate how it could be playing a role in sending signals to the brain and modulating aspects of a variety of things, including interoceptive signals, and to help us feel how we feel. And So that's the large component, which is a neural one. And then on top of neural component, we have a hormonal component. So we have lots of cells in the gut that release hormones, intraendocrine cells. And these can either affect the neural signals, or some of them can also affect other targets to get to the brain. And so this has been very much studied in the context of feeding behavior, because feeding we need a system in our body to tell us when we're hungry. And so we need to understand within the gut-brain axis that there's hormones being released in advance of hunger, and also hormones being released to tell us to stop eating. And these are evolutionarily wired. So that's a second component of the gut-brain axis. The third component of the gut-brain axis is the immune system. And we've all got very familiar about our immune systems in the last 2 years. But immune signals, our gut is the reservoir of some of the highest levels of immune cells, and they traffic to different places, and they're able to become activated in response to certain injury or infection or various systems, and are able to help communicate across the body and even into the brain overall. So they're the major what I would say components of the gut-brain axis. And then on top of that, we have the microbes. Microbiome then can interact with each of these pathways in different ways, and complicated ways that we're still trying to really work out to influence brain and behavior.
Interesting. So this enteric nervous system, you said that there's actually more neurons in this nervous system of the gut than in the spinal cord.
Yes.
And my gut instinct would be that they're mostly involved in moving the food through, causing the movements that actually
peristalsis and motility in general, and that's what they're one of their main fashions their main roles are. But it also plays a role in things like abdominal pain. When you get cramps, and you get if you have irritable bowel syndrome, which is a disorder we study, which is a very unloved disorder. No one wants to talk about the Center for irritable bowel syndrome. We don't have donors lining up to give us money to do that, but it is a very common and disabling disorder, which has the enteric nervous system at its real foundations in terms of what's going wrong and how that pain is processed. It's processed through spinal mechanisms into the brain, and so everything is connected.
I see. Interesting. So the enteric nervous system is not only moving the food through, but it's doing some kind of pre-processing that then gets shuttled up to the brain.
Yes.
Interesting. One of the things that I wanted to talk about is just a general area of discussion is the connection between the microbiome and this gut-brain axis and aging and immunity. And I know that you and others have done experiments that center around something called a fecal transplant. And the basic idea is you put a young mouse's microbiome into an old mouse, and interesting things happen. So very broadly speaking, how do these experiments work, and what are some of the high-level changes that you guys observe with respect to aging and immunity?
Okay, no, great question. So I'll rewind a little bit just to give listeners a little bit more context about why we would think about doing this in the first place. Mm-hmm. And so what my lab is really interested in doing is understanding this relationship between the microbiome and the gut-brain axis at key windows across the lifespan. So we have programs in early life, in the perinatal period, in adolescence, and all the way into old age. And buoyed by experiments in humans that my colleagues here in Cork did, it's almost a decade ago now, where they showed in a project called ElderMet that elderly individuals who tended to live in nursing homes and assisted living, that their microbiomes had a reduction in diversity as they aged. And this correlates frailty and other health outcomes. So the people who lived in the community then, who had very much more diverse microbiomes, had less frailty, and and they went one step further to show that it was driven by diet. So the people with the more diverse diets had the most diverse microbiome and were less likely to be frail. Now, so that was kind of interesting, but I'm a neuroscientist, so I was a bit disappointed there wasn't a lot of neuroscience in that study, even though it was in Nature, but it wasn't driven from a cognitive or neuroimmunology perspective. There was a peripheral immune measures, but we were kind of excited about it, and it reminded us of Eli Metchnikoff's work at the turn of the last century. Now, I don't know if you've heard of Eli Metchnikoff, Nick. Okay, no. And Eli Metchnikoff is, it's kind of timely. He was a Ukrainian Russian working in Paris who won the Nobel Prize in 1908 for discovering the process of what we call phagocytosis, so the gobbling up the immune cells do of foreign bodies, and so he was a brilliant father of immunology, brilliant guy, and maybe as happens quite a lot of great scientists later on in their careers, they can start to come up with crazy ideas. And Metchnikoff was really full of crazy ideas. Most of them complete nonsense. But one of the things that he when he turned 50, which at the turn of the last century was you were getting old. He started to get worried about aging, and he started getting worried about his own mortality. And he really was getting, wondering why, how can I do something about this? And so he started looking around in populations where people seemed to live longer and healthier. And this brought him to parts of what's now rural Bulgaria. And what he found was that people who lived there that lived longer, ate a lot of fermented foods containing lactic acid bacteria. So he put forward the idea over 100 years ago that it was healthy aging could be driven by lactic acid bacteria, and is really the father of probiotics. And, if you go to Korea, you can get a yogurt drink with Mechnikov's picture on it. So, it must have that Nobel sign of approval. It must be good for you. But anyway, as I say, most of what he talked about was nonsense, and so he was ignored. And most of this was ignored for 80 years. But you can read about it. He wrote a book about it, about the optimistic studies on aging, it's quite interesting, because sometimes in science we are just reinventing wheels that have been turned already. We're inventing them with technologies now that can advance that. So Mechnikov then is the father of lactic acid bacteria and probiotics. And so he's well known if you're an immunologist or a microbiologist. I'd never heard of him until I started reading these studies as a neuroscientist. But buoyed by the studies from my colleagues here in Cork on the people in the nursing homes, and with this Mechnikov idea, we started I wrote a paper called Revisiting Mechnikov, which was really about looking at using aged animals, mice in this case. We looked at what's going on in their brains, what's going on in their behavior, and how is that linked to what's going on in their microbiome. And we found quite a number of what I would correlations. But it's just correlations. Mm-hmm. So it matched kind of what we were seeing in the human studies, that the microbiome seems to correlate with some of the behavioral changes and with some of the brain changes. But we needed to go one step further. And so in a study that we followed it up with, we said, could we target the microbiome in aging, and through diet would be the best way? And could we slow down some of the effects of aging? And so we used a diet that was enriched in inulin. Now, inulin is a fiber present in lots of vegetables, chicory, leeks, artichokes, etc. And for these studies, then I turned to middle-aged mice, because I started getting very interested in the middle-aged brain myself for obvious reasons. And there already in middle-aged mice, you start to see neuroinflammation happening in the brain as an harbinger of later, perhaps degeneration. And so what we found was when we fed the animals this inulin-enriched diet, that they didn't have this neuroinflammation in their brains. And that was really cool, because now we see that by feeding microbes, we could actually slow down the effects of aging. But it wasn't definitive in terms of the diet could be having direct effects on the brain independent of the microbiome. How do you know? How did I know? And I was asked this question many times. How do you know it's the microbes? So that led to the study that you're alluding to, which was this kind of killer study that we wanted to do, which was kind of a proof of concept. Well, what if we took the microbes from young animals and gave them to older animals, which have a different microbiome? Could we reverse? Now, this was the real killer part of it, because we went to old animals, 22-month-old animals. Could we reverse the effects of aging on brain, behavior, and immunity? And that was really, an experiment that I told the postdoc, this is not going to work. We have to do it. And so we took advantage of fecal transplants approach, which you alluded to, which is exactly what it says on the tin. It means taking some other individual's poo and transplanting it. In this case, it's from one mouse to another. And I'll just stall and just explain to your listeners what fecal transplants have got some attention in the press over the years, and they're seen as something quite new, but they go back to ancient China, where they were used in the treatment of all sorts of ailments. It was called a yellow soup in Chinese medicine. And they've been revolutionary in modern medicine in treating certain forms of infection, most notably Clostridium difficile. C. diff kills people, and it's antibiotic-induced infection. And what studies from the Netherlands almost 10 years ago, again, showed that there was a 95% success rate in individuals that had been given a fecal transplant for this infection. And so I work in a medical school. There aren't that many areas of medicine where you can say there's 95% success rate for anything. So that brought fecal transplants back into the mainstream in humans.
Wait, so now, I just want to make sure I'm understanding. So I've been asked this before and I don't know the answer, so I have to ask. How are these transplants physically conducted?
Oh, yeah, sure.
And then you're saying that in this particular example, there was some sort of antibiotic effect?
So no, in the human studies, what happens? They're used to treat predominantly Clostridium difficile, which is an antibiotic. It's caused by overuse of antibiotics. It's an infection that becomes resistant to antibiotics, and so your antibiotics don't work. So it basically will kill you in hospital. It's basically a hospital-acquired infection that people die. All over the world, people die today from it in Western world. And because it's caused by antibiotic and you try and treat it with antibiotic, you reach a stage where, something has to change. And what you do then, what the studies with the fecal transplants do is, well, what if we can get rid of the microbes and put new microbes back? And will that work? And so in terms of the physical purpose of doing it, there's really only two ways you can get to the colon. You can either go down in a nasogastric tube, or you can go up in an enema, and that's how people deliver this overall. And but the success rate was just dramatic, and now it's been tried in all types of different illnesses. There's ongoing programs in depression, in anxiety, also in obesity, in inflammatory bowel disease, and irritable bowel syndrome. Mixed effects. It's not going to be the cure-all. What a lot of people are also looking at is, well, could we come up with a more innovative way of delivering this disruptive medicine? So there's products like crapsules, which are basically poo that's been put into pill format. And this is gaining a lot of attention from an innovation perspective.
But is it is it like a purified preparation of some of the bacteria?
No, well, you can do that, and some groups are doing that. So there's a group in Arizona that are doing that with the consortium of about 100 bacteria. What people are really looking at is just from donor, from fresh. It's remarkable. Interesting.
So the idea is just you're putting in other bacteria that then outcompete the one that you don't want there.
Yeah, exactly. And they can grow over, and they can, whatever else. And so in the C. diff world, in Clostridium difficile work, it works. And in outside of that, it's more equivocal, shall we say? Yes, but we need more studies. But with that knowledge of clinical medicine and how this is working, that fecal transplants are moving into the mainstream in such a way that almost all Western hospitals nowadays will be performing fecal transplants regularly, then it becomes a more viable approach to use in our animal models as well, because it has a translational potential overall. So that led us then to think, well, could we do this in our aging animals? Could we take the microbes from young animals and give it to the aged animals and reverse the effects of aging? And quite remarkably, and to my surprise, many of the effects we had seen were changed at the level of the gut, at the level of the immune system, at the level of the metabolome. So that's the chemical makeup of the region of the brain called the hippocampus, which is involved in learning and memory. And we were able to reverse some of the age-associated behaviors. And so we published that in Nature Aging. We got on the Steve Colbert show joking about it. He made jokes about our studies at the time. It was kind of one of these papers that had got a life of its own. It was really important to show the next steps from our earlier work, where we could go, but it asks so many questions.
Mm-hmm.
And before your listeners all try and rush out now and start stealing the poo from infants, that is not what I'm saying. And that's not where this research is really advocating, really. What it tells us is that it gives us some really good evidence, at least in a mouse, that the microbiome needs to be looked after as we age to engender healthy aging, to engender our brains for healthy aging. And, we need to look at ways of doing that. Mm-hmm. And so that's kind of, where it's at, rather than the fecal transplant was a proof of concept.
Mm-hmm. Another way I could have imagined exploring the necessity of the microbiome for some of these diet-induced effects would have been to give the mice what you mentioned earlier in terms of diet, but sort of nuke the microbiome with antibiotics first. Has anyone ever done something like that?
So yeah, not the same way in aged animals that I'm aware of. We were thinking of doing that, but we were a bit worried that aged animals are very expensive, and they may not respond to the antibiotics are quite a, there's a tolerance effect. So that needs to be done, and that would have been the cool experiment that we're still on our list of things that we should do. But there's going to be other collateral effects of antibiotics in the gut in terms of energy absorption and food, that's going to complicate things. Mm-hmm. That could give you indirect effects. So the way we did it, I think, was probably the most indicative that it is actually the microbe, because the only two differences between these two old animals is that one has got the microbes from a young animal, and we gave the other the microbes from an old animal, from an old animal. So we were able to, control for the transplant itself.
And so when you do this, the old mice have brains that start to look younger, in effect. Yes. And so what does that actually mean? What are some of the markers that differentiate the younger looking versus the older looking brain?
Yeah, so we looked at a variety of different ones. So the really intriguing one for me was the metabolome in the hippocampus. So this is using mass spectrometry, to look at what are the chemicals in the brain that are being formed, and there are hundreds and hundreds of chemicals, and we were able to use this approach then to see which ones were changed with aging, and then which ones were reversed. And there were quite a lot that were sensitive to the fecal transplant. And so that was kind of really cool, because some of these may originate in the gut, because one of the things about the gut microbiome is that it's like a little factory producing all kinds of weird and wonderful chemicals that our bodies wouldn't have otherwise. And some of these chemicals can get into the circulation, get across the blood-brain barrier and into the hippocampus. So that was quite intriguing. We also looked at microglia. Microglia are the brain's immune cells, and we found that as the brain ages, there becomes an increased activation of microglia, and there was a diminution of this when we gave the transplant. We did look at other things that didn't change. So not everything changed. So we're really interested in a process in the hippocampus called adult hippocampal neurogenesis, which is the birth of new neurons that occurs throughout the lifespan. And, it starts to get less and less as we age. And so we found the age effect, but it was still there. The transplant wasn't able to recover these new neurons to be more. So not everything changed, but quite a lot of things did. And then on behavior, we found that when we put the mouse in a maze, in particular a Morris water maze, so it's basically a spatial learning task, they were able to find the hidden platform and develop a strategy, again, similar to younger animals, when they had been given the transplant. Mm-hmm.
And I suppose one of the things that's interesting here is the fact that you see this effect means, at least for some of these things, because you're able to reverse some of these inflammatory markers and even behavior and things like that, it's not like the aging process has just sort of accumulated over time, that some of the stuff actually is acutely reversible. That seems surprising.
Yeah, I know, and for me too. It reminds us that the brain is so plastic. It reminds us that there is so much potential for this, and it reminds us that it's never too late. to focus on doing these things.
Mm-hmm. And when you do these microbiome transplants, so in this case, you're going putting the young microbiome or the microbiome from the young mouse into the old mouse, you see these remarkable effects. Does that new microbiome, is it stable or does it revert passively back to the old version?
No, a great question, and we don't know. We didn't do them experiments yet. Getting aged mice costs us a fortune because we don't have a National Institute of Aging here in Ireland that can deliver these to us. And it is an important aspect to know. In terms of, do you have to keep doing? We did give them boosters twice a week during all of the testing that was going on. So just to ensure that we got some engraftment continuing.
Mm-hmm. And when you look at the mice, do they look healthier just visibly?
No, not really. Not really. We didn't see a huge. They are quite healthy, and it's one of the functions of working with aged animals is that you work with healthy aging animals anyway, because if the animal starts to deteriorate, you have to for humane reasons, you have to sacrifice when you start seeing anything going wrong. So they weren't that visibly aged. They weren't. Unfortunately, they had no walking sticks or glasses or anything that we could look at that would be able to tell them, or they were going for early bird menus or anything,
And so, what about effects in the periphery, in the enteric nervous system, or inflammation markers there?
So again, peripheral immune markers, we did a whole panel of different peripheral immune markers, and it was somewhat selective, but it was really there that a lot of the aging or induced effects were sensitive to it, and that makes sense. And we have this process that's been coined inflammaging, and it's basically as we age, our immune systems, and that's why during the pandemic we were so focused on keeping our vulnerable people over 80 initially and then down to over 65, because their immune systems are just not able to tolerate things. And so we were able to push back some of these signs of aging in the peripheral and locally in the gut immune system as well. So that was kind of cool. Again, we don't know what's driving what first. We don't know, is it the immune? If you reverse the immune, is that what's causing the knock-on effect? And we'd like to dig into the pathways that might be at play here. And we really need to invest in more effort in that.
One of the things here that's obviously interesting is you're seeing these effects at all, and it goes all the way into the brain. Stuff is getting all the way into the brain and having behavioral effects. I know that there's also been some work linking the microbiome to social behavior in animals. So, starting wherever you think is most appropriate, what has the link been so far between the microbiome's ability to modulate social behavior?
Okay, so this is a really intriguing topic, and one I'm quite excited about in terms of where we could go with this. In 2013, so it's almost, we showed in mice that lack microbes. So let me rewind. So I've been talking about this microbiome gut-brain axis for quite some time, and my neuroscience colleagues are a good skeptical bunch of people, and so I can talk about correlations and I can talk about gut feelings and I can talk about things, but they really they want mechanisms and they want more evidence that something is, that the microbiome. So a decade ago, we first published in 2011 showing a specific strain of bacteria could have effects on brain and behavior through the vagus nerve, and that got a lot of attention. And it was kind of one of the breakthrough moments for me in terms of really getting good evidence that this was relevant. But it wouldn't, we needed more data. And so how do you get more data that something is important to a process? Well, you take it out and see what happens. And so that's why I always say to my neuroscience colleagues is that here's some really hard data. If we take microbes out of mice, and we have a germ-free facility here in Cork, and we look at the brain and behavior of these animals, then we start to get an idea of, is the microbiome involved, yes or no? And early work out of Japan showed that the microbiome was involved in stress response. Work from Karlinska from Rochelle STS Heights and from Jane Foster and McMaster and our own group showed that brain development and variety of brain processes were gone awry in these germ-free mice, especially in the context of anxiety behavior. And then in 2013, we looked at social behavior, and we found that mice that are growing up without microbes have clear deficits in both sociability and social cognition. So sociability is basically when we give the mouse the opportunity to spend time with another mouse or an object, a normal mouse will gravitate towards a mouse. They're social, they want to spend time, but not a germ-free mouse. Also, then for social cognition, we want the animal to be able to distinguish between its everyday playmate and a new playmate. And mice will gravitate towards a new playmate. So they're, fickle, like maybe some humans, but they are quite fickle in that regard, but not if they're germ-free. And so this was really important data for us to show at the time that if you're lacking microbes in your gut and you're a mouse, that you're having different social experiences overall. So that really got me excited. And we did a lot more digging into the mechanism of this. We showed that in these germ-free animals that they have deficits in their amygdala in terms of the arborization of the amygdala, which is key brain area involved in social behavior. In the prefrontal cortex, which is also very much part of the social circuit, we showed that there was increased myelination. And people think about myelination in the context of multiple sclerosis, where it's a demyelinating disorder. Well, here we got increased myelination. We also get changes in the hippocampus, in arborization, and in neurogenesis. So we're beginning to build a circuit-based approach to understand that the brains of these animals are pretty messed up, but they have these social deficits. And then when we put the animals in social contact, we also looked at using unbiased RNA-seq approaches. We looked at the transcriptome and were able to show how different activation of a variety of key molecular pathways were underpinning social behavior were also gone awry in these animals. So, and around the same time, Elaine Chow's group, then at Caltech, now at UCLA, showed, again, social deficits in animals that are being given early life infection with the maternal immune activation protocol. And this correlated with the microbiome changes, and she could reverse this with a specific probiotic. So again, the social interaction and she was able to cite our paper in that, and so that was really cool. And then since then, I'm just really, I want to know more about why just the social brain is so sensitive. So two things that I learned on this journey. One is that you can't do everything yourself, and you need to collaborate with experts. And experts get a bad name, but it's good to have experts. And so I had a very great postdoc in the lab, Roman Stilling, and when we got this data on the social brain, then we started reflecting from an evolutionary perspective about why would you need microbes in the lumen of your gut if, for normal social interactions. And so we started building a kind of a framework based around the cortical development theories of Robin Dunbar, where he showed that social, the elaboration of the cortex is greater in social animals, maybe driving social behavior. So we know that social behavior is driving brain development. We know the microbiome is driving brain development. We know the microbiome is changing social behavior. So is all of this just all linked up in some way overall? And then we start thinking about, okay, and this is before pandemic, before people were much more attuned to this. But if you're in a social environment, your microbes are getting passed on much quicker to other people. And so I started thinking about, well, what's in it for the microbes? And Rome and I were going to write. We were writing something on this, and then I collaborated with a guy in Vanderbilt, Seth Bordenstein, and Seth is Howard Hughes there, and is an evolutionary microbiologist. And I put some of these ideas to him, and he basically told me that I was completely wrong to even be thinking like this. And every now and then, as a PI, it's really good to be told you're wrong. And why was I wrong? Well, I'm wrong because I'd forgotten, something that's very fundamental to this whole story, which is that the microbes were there first. Our brains have never existed without microbial signals. Our mitochondria in our cells are just microbes that got lost. So we need to really reframe how we think about the gut and the brain as if they're two separate things completely. And this is the problem with modern medicine. We love to compartmentalize the body, but there's never been a time where brain development hasn't had these signals coming from it, and they've evolved into allowing for appropriate social behavior. And there are friends with benefits and are friends with social benefits. And so we wrote that article, and it was kind of a cool idea, and it led to a lot of other thinking. Then you start to look across the animal kingdom. And then I collaborated with a zoologist, John Quinn, here, as well as with Seth. And we started looking, well, if you start interfering with social dynamics or hierarchies across the animal kingdom, one of the things that keeps coming up is that you change the microbiome. So whether it's in a bumblebee in a hive or a baboon in the wild, these data exist, and vice versa. If you start to change the microbiome by either diet or other approaches, from insects all the way to non-human primates, we begin to see that the social behavior is the one thing that seems to be quite sensitive to this. So there's something about the social brain, and that makes it sensitive to such signals. And yeah, I haven't talked about disease on purpose yet, but that's where it goes a little bit. But I want to, before I go into talk about disease, I want to talk about normal social behavior. How much of our decision-making, how much of our social decision, how much about how we eat, and how much of our teamwork, our altruistic behavior, our cooperative behavior, our empathy, our competitiveness is all driven by what's going on in our gut and how it's speaking to the brain in terms of normal behavior. Then when it goes wrong, what happens?
So how do you start thinking about that normal behavior stuff? One of the places where my mind goes naturally, having a background in evolution and behavior, is, if the microbiome is inherently tied up with things like the state of not only your metabolism, but your immune system, then, the compounds and the markers that a conspecific can perceive, which are connected to those microbes, is going to contain information about the general physiological state and health of the other individual. So that might be used in terms of mating behavior. That might be used in terms of avoidance behavior, if you want to avoid someone who's potentially contagious or something like that.
Absolutely. I mean, social immunity is really important in this. And so we put all this together. And if your listeners are interested, it's in a review article we wrote in Science probably 2 years ago now, because we wanted to capture these exact same things. And of course, insects and mammals in the wild will have a different need for sensory. And so there's a sensory component to social, and some of these chemicals that are coming from bacteria can have really olfactory or various other cues to allow it. But I guess the main thing, Nick, that I'm trying to get across is it's all connected. It's all connected. And the microbes were there first. And that's really, my mantra. Some of this, when I try to explain to people, people always think it's something completely different. And what we don't know yet, and this is some of the studies I want to do, is really if we took say 1,000 people and stratify them based on their social decision making within a normal range. Could we look at what's going on in their microbiomes in terms of driving that, and then could we change that through diet?
Yeah, I mean, and it strikes me too that animals are effectively sampling each other's microbiomes, or at least portions of it, in terms of the behaviors they display. Like, for example, if you just think about like affectionate behaviors that humans have, I mean, like why do we kiss each other? I don't know the answer to that or how it evolved, but you are going to be sampling a portion of someone's microbiome, and it probably contains real information about their what you might just think of as their physiological compatibility with you.
Well, people are looking at that, and indeed, one of my colleagues in Germany, that's one of her areas, is looking at how couples interact. There's been studies on houses, when people move houses, and how their microbiomes change, and how when people start going into relationships, and when people all of this is becoming. We're getting more data. And it's going to be more complicated than we think, and it may not be as I guess the relative contribution of it to specific behaviors versus other environmental factors or lifestyle factors that could be overriding some of this in the normal basis it needs some form of investigation. But I do think, when we talk about the social brain and microbiome, we do need to look a little bit more at normal aspects of social life. And I think and some of the work in the honeybees is just phenomenal, because they're such a social insect, and if you do, some of the problems we're seeing in the world with pesticides and various other things that are impacting the microbiomes and the social behavior and various things. What are some of the salient results from the honeybee work? I'm not familiar with it.
Yeah, no, it's just this accumulating data now showing that if you mess up with the microbiome in honeybees, you mess up with social behavior. And if you mess up honeybee social behavior, it has a knock-on effect on biodiversity because of what they're doing, et cetera. So it's building it into a planetary health framework that you wouldn't even interpret beforehand. And what's disturbing the microbiome? Well, a lot of the work is experimental right now, but we do know that certain pesticides and various other things can do that. And there's a whole environmental aspect to that.
I mean, based on that and based on, the experiments you described using antibiotics in mice, one naturally wonders what the effects of things like antibiotics in the food supply chain are having on humans.
Yeah, and this is real, especially in the US, because there's a higher tolerability for antibiotics within the food chain in the US than there is in Europe, for example. But Marty Blaser, who's now at Rutgers, has written extensively about this in the context of metabolic health. And he's data in animals, and then he's human data. He basically thinks that the obesity epidemic is driven by early life exposure to subclinical levels of antibiotics overall, and that we're missing microbes overall. That was the title of his book about this. But he also shows, and the correlations of antibiotic prescription rates in the US on a county by county level, and it maps over the obesity levels like almost verbatim, And so there's become a bigger appreciation of the importance of the microbiome in all aspects of health, particularly metabolic and cardiometabolic health. The other thing that's informing this is studies where people are going out and looking at the microbes of ancestral tribes. So going to Tanzania, and there you can see the microbiome is very diverse, largely driven by the hunter-gatherer lifestyle that they have. The gathering part is often neglected in this. They have to eat a lot of fiber and a lot of grains and a lot of things. And so that's, and then if you go to more agrarian communities like in Malawi or Venezuela, you see what introduction of farming has done to the microbiome, and you start to see a diminution. And then in our Western world, whether it's in the US or here in Europe, with the introduction of processed food,
the increased stressful lifestyle, the antibiotic exposure, the environmental pollution around us, we have an extinction of these microbes that our ancestors have. And work from the Sonnenbergs, Justin and Erica Sonnenberg at Stanford for some years now have been really highlighting how this could have long-term consequences, because we don't know if we can get them back. Is what's gone. And, you can eat as much fiber or whatever good food as you want. If you don't have the microbes there to actually get the, good stuff from it, then it becomes a problem. And, but that enables us to start looking at, so they've done studies on these, microbes and being able to link it to overall health.
Mm-hmm. I think, a skeptic might say, well, of course, a hunter-gatherer's microbiome is different. Virtually every aspect of their physiology is probably different because their entire lifestyle, from diet to activity level, is different. But to connect it with some of the experimental work you talked about at the beginning, we at least have rodent evidence that a transplantation of microbiomes can be causally related to at least some changes in physiology.
Absolutely, and that's where we want to go. And what our lab is doing is, like, a lot of the work is being done in metabolic health or immune health and gut health. We want to bring it to brain health, and that's really our the different perspective that we have on this overall.
Now, I think if there's more to talk about normal animal behavior and its relationship to the microbiome, we should definitely do that. But some of the results that you were describing with respect to social deficits naturally make one think about things like autism.
Yeah. So, but we also, and I'll come on to autism, and I have no, but we also should remember that there are other disorders of sociability besides autism. Mm-hmm. Social anxiety disorder is something that we're studying right now a lot, and are quite intrigued on, and it's a fascinating disorder, and it is really more common than people think it is, and we have new data to show that the microbiome is different in people with social anxiety disorder. Social deficits are a core symptom of schizophrenia, and really are quite hard to treat with antipsychotic medication overall. Social disorder, social anxiety aspects can be at a subclinical level with general shyness or general social awkwardness overall. But let's go to autism because it is probably one of the most studied areas in relation to the microbiome. It also is one of the most emotive and one of the most that can be the most challenging to discuss and manage expectations of individuals overall on it. My take on it so far on the data where we are is that there seems to be some very clear relationship between the microbiome in early life and the natural history of certain symptoms of autism. Now, the early experiments in this field were coming from, for example, Sidney Feingold's lab in UCLA, where they showed that vancomycin as an antibiotic might have some beneficial effects in autism. They did some very small scale human studies, promising. There's been lots of anecdotal data, somewhat promising. And then when people started measuring the microbiome in a compendium-like way, just what microbes are different in autism versus not. The early studies were largely small, heterogeneous, didn't really take into account diet, were a bit of a mess, to be honest. You couldn't really interpret anything. Yeah, there's changes, but what do they mean and are they consistent? And I recall, it's probably almost a decade ago or a good number of years ago, looking at one of these better papers in the autism field that was emerging to show that there were links between microbial changes and a subset of individuals in this larger study with autism. And but the good thing in the study was they had very good dietary data. And what the dietary data showed was that it wasn't that there was a link between autism and the microbiome, but it was a link between what these kids ate, and they ate a lot of chia seeds. Now, I didn't even know what a chia seed was, but chia seeds are like a superfood that are often in smoothies. and used a lot by autistic kids and their families. So that was just a clear example of we need to be very careful with this field about where it's going. Where things have got exciting is work coming from Arizona, where they've shown using a kind of a fecal transplant approach that I alluded to, but they developed a consortia of bacteria, and they showed in a small enough study some years ago. This is Rosa Kramick-Brown's work, where she's really along with Jim Adams. They showed that when they gave a fecal transplant to a group of kids with autism, that their gut and their social problems resolved somewhat, and that this persisted up to 4 years. So that was kind of it's a small study. It's not placebo controlled. It has issues, but it is exciting. And then the animals from that, Sarkis Mazmanian's group at Caltech took the microbes from these infants, or they were now children, and then showed that they could induce autistic-like behaviors when they transplanted them into mice, germ-free mice, and allowed them to breed for one generation. Now, this study's got some kickback because people felt that they were over-interpreting some of the statistics and over-interpreting some of the things. But, there was lots of really intriguing data in this paper overall. It did show that the microbes from kids with autism induce different effects on brain, on behavior, on the metabolome, on immunity in these compared to those from neurotypical kids. So for me, it was kind of exciting overall. And then just 2 weeks ago, Sarkis's lab showed again, based on our work on myelination and others, they showed that a metabolite from that was identified in these kids could also induce autistic-like symptoms in mouse behavior. And the question is, how real is social behavior in the mouse for humans? Mm-hmm. And then they had a proof of concept paper in Nature Medicine showing that a new compound that it basically mops up these metabolites, that basically quenches them in the gut, was having beneficial effects. So the story is really there. Oh yeah, and then I will do my one minor pivot, just because in the interest of clarity, is a large study from Australia that came out about 3 months ago, where they looked from a genetics group there, which looked at the microbiome in really good detail. It's a really good characterization study, and they looked at food intake, and they looked at food behavior, and they looked at different aspects of eating. And what they showed that the microbiome changes in these kids was driven by picky eating in these kids, and wasn't specific to the actual behavioral changes per se, but so the press interpreted this, and the press releases allow them to interpret this, that there's no relationship between the microbiome and autism. But it's actually more nuanced, because we don't know what led these kids to be picky eaters and what led their neurodevelopmental trajectories. And so it is complex, and it is gene environments, microbiome and gene interactions that are going to be at play. But we need to kind of understand this in a different way. So our research is very much looking at early life microbiome changes, and we've shown this now germ-free in animals born by cesarean section, because C-section changes the microbiome, and by giving antibiotics, maternal antibiotics to mice. All three of them, the one behavior we see that comes out of time and time again is social deficits. So there's something about the social brain. So why wouldn't a disorder like autism, which is very spectral, very distinct, why wouldn't it be also involving how the microbiome is shaping, talking to the brain at key critical windows?
Interesting.
I hope you didn't want a short answer.
No, it's fascinating stuff. So you mentioned, autism is just one form of social disorder. And there's many other afflictions that people have that might be related to the microbiome, I suspect. So when we think about things like social anxiety and depression, what are we starting to learn there?
Yeah. So in social anxiety, we are finding that there are distinct changes in certain microbes, and we're getting this ready for publication now, and we're doing the experiments in animals to look at the mechanisms and underlying that. In depression, it's a little bit complicated, again, because a lot of the early studies are heterogeneous, using not the best tools. But we were really interested in this some years ago, where we wanted to see what if you took a bunch of people with clinical depression. And this was in collaboration with my colleague, Ted Dinan, who was the chair of psychiatry. Clinically depressed individuals versus healthy individuals, took their microbiome. What we found was there was a lack of changes between them. It wasn't a big number, so all the caveats with that. And then there was a lack of diversity, a reduction in diversity in the depressed individuals. So kind of like almost accelerated aging in their microbiome, if you want. What we did is we took the microbes and we transplanted them, as we're prone to do, in this case to rats. And much to our surprise, the rats developed depression-like symptoms. So this was kind of really cool because it helps us with causality. It helps us really answer, Now, how do you know a rat's depressed? Well, that's a really hard question. But we look at key endophenotypes of depression. So anhedonia, the ability to experience pleasure. Microbes, when the rats had taken microbes from depressed people, they had less interest in sweetened sugar compared to normal animals. They were more anxious. When you put them on a maze where they can go and explore open arms or stay closed, they stayed in the closed arms. We looked at their inflammatory profile, and we know inflammation is also present in depression, and we were able to change completely their inflammatory profile. So it's remarkable. And this has been reproduced by a group in China. It's been reproduced by a group in Canada. Chimik Burchik's group is working on this in the context of anxiety. And so, it really helps us develop a kind of a causal relationship. So that's one of the things. The other thing, and that's happened in the depression field, is work from Belgium, where they looked at 1,000 people. So that's good size to start with. So, because one of the big problems here is all about sample size. Mm-hmm. And they looked at those that were depressed, and they found that they had a specific change in their microbiome. They talked about enterotypes, which is kind of just subtyping the microbiome in different ways. And they went one step further to show that the potential of that microbes to make certain neuroactive chemicals that could potentially get to the brain. So they were able to identify which bacteria make which chemicals. And they were able to show in depression that there was a reduction in certain bacteria and an enrichment in others. They were able to verify these data in another 1,000 people in Netherlands, and then in a very small sample of really depressed people in a hospital in Belgium. So that's kind of neat, and we're collaborating with them to do more mechanistic work on where this is going. And I think what we're beginning to see really is that the microbiome could be targeted for mental health benefit. And then that's why we coined the term here in Cork. We coined the term psychobiotic, which is basically a way to target the microbiome for mental health benefit. Not to be confused with your psychedelic, but psychobiotic mm-hmm. is something that we're very interested in.
Well, so there's something really interesting I'm thinking about here, which is, you've mentioned, everything that, when I think about everything that you've mentioned, the microbiome can have, it seems, effects on the brain through a variety of different pathways. We've been talking about things like depression, anxiety, and these things that involve psychoactive effects. There are, as people may know, there's a lot of serotonin used in the gut, in the enteric nervous system. And I wonder if, we've got anything from selective serotonin reuptake inhibitors to serotonergic psychedelics often have in people gut-related side effects. And I'm wondering if the microbiome itself can be affected by some of the psychoactive drugs that people are consuming by the millions.
Absolutely. We've done these studies in animals and characterized it well. Initially, we were interested in antipsychotics. So atypical antipsychotics are widely used. 1% of the population in the Western world are on them, which is quite a lot.
Mm-hmm.
And they're not just used to treat schizophrenia, but they're used to treat many aspects of depression and bipolar depression as well. So, but one of their side effects is metabolic dysregulation and obesity, and people just balloon. And so we were interested in looking at, could this be driven by the microbiome or the microbiome-gut-brain axis? And so we showed in 2013 that olanzapine was mediating its effects through, that was affecting the microbiome. We followed it up with a paper where in antibiotic targeted animals that this didn't happen as much. So, we're really getting to that level. And then Sofia Cosoto, when she was a PhD student in the lab, looked at seven or eight other psychotropic drugs, so including mood stabilizers and SSRIs, and showed that they had quite a profound effect on the microbiome. And just as we were getting that ready for publication, a paper from Germany came out in Nature with over 1,000 drugs in it. But it showed very clearly that a quarter of all drugs in pharmacies today are having deleterious effects on your microbiome. And in the drug class they highlighted was antipsychotics. So it was really cool. In relation to psychedelics, or that, I don't know, is there that much data, but I know people have put forward this idea. Kim Kuiper's lab has talked about this in some of her reviews.
So that's super interesting. A lot of the pharmaceuticals that we're using are affecting the microbiome. It's not so surprising that that's happening to some extent, but it's maybe surprising that it's happening so widely. So just to give people a more concrete sense of this, you're saying you've done experiments where you give rodents an antipsychotic. There is some sort of downstream metabolic effect, and the size of that effect is diminished if the animal is taking antibiotics.
Yeah.
And what are those effects typically for like an antipsychotic?
So it's weight gain, it's accumulation of fat depots. It can lead to diabetes at a really high level, but in our animals we don't get that far. It was really surprising data, and it's really a clinical problem. But now there's a whole area of research called pharmacomicrobiomics. So looking at, I trained as a neuropharmacologist. So I'm interested when I would teach, I would tell students that you need to think about pharmacodynamics and pharmacokinetics. Now, if I'm teaching, I say you need to do pharmacodynamics, pharmacokinetics, and pharmacomicrobiomics, because that's really important to how drugs are impacting the microbiome and how the microbiome is impacting drugs. Because we just also published a paper showing that the bioavailability of this antipsychotic olanzapine was also affected if we wiped out the microbiome or we gave a probiotic. So there was differential effects there. And it's really exciting. Again, not so much yet in neuroscience or in psychiatry and neurology field yet, but in oncology. Right today, people are starting a treatment for melanoma will take checkpoint inhibitors. The efficacy of them checkpoint inhibitors will be dependent on their microbiome composition. And that is really, really dramatic. And so that the question we're also interested in, this is work my colleague Jared Clark and I are doing right now into the future. We're looking at the side effects of some of these chemotherapeutic agents, brain fog, cognitive changes, social effects. Could these be driven by what these drugs are doing on the microbiome? And it's opening up a whole new potential in that regard.
And what was the number that you cited for the percentage of drugs in a typical pharmacy?
Approximately 25% of a quarter of all drugs in pharmacies in this paper where they looked at 1,000 drugs. Now, this was all in vitro, so it wasn't, we were doing our stuff in animals. They just did, but they went for we had 9 drugs, they had 1,000, so they went on breadth overall. And it's really exciting to think that, but it's also worrying. So the question is, for any drug now, is its side effects could be due to collateral effects on the microbiome, or could its efficacy be enhanced by, and so understanding, and this is also relevant to foodstuffs, because foods most of them are chemicals, not a million miles, you know. So if you take polyphenols which are all the color in your vegetables, if you take them into your diet, they get broken down into flavonoids and then into other, and they get broken down by the microbiome into other chemicals that can have other effects. And so you know, it is something that's receiving a lot more attention now. We're also interested in environmental chemicals like pesticides and what they're doing and how they're being taken in and how they're been acting on the microbiome, and could that have a long-term effect overall? It's as I like to remind people, everything is connected, I guess.
It would be so it'll take me a second to build this up, but it would be shocking to me if there weren't some drugs that weren't directly metabolized by some microbes that can be in the microbiome. And I wonder, you know, if there are any clear examples where, say, you give drugs to a group of people, or you give drugs to a group of animals, and if there are multiple cohorts of animals in terms of microbiome composition, are some of those drugs being made active by some of those cohorts? So like anything like that?
Oh yeah, no, but people have been using prodrugs for years so that they can be broken down in the treatment of IBD and various, a variety of drugs that they use microbial metabolism as part of their delivery device, because you wait till they get to the small intestine or whatever else, and so that is already happening in pharmaceutical sciences. Interesting.
So I mean, that would imply that sometimes when a drug does or does not work, it may or may not be because of the drug per se, but because, you know, maybe the active ingredient wasn't there in some people because they didn't have the microbes for it.
Yeah, it could be. And that's definitely what's happening in some of these cancer treatments now.
So what is that?
So like with these newer checkpoint inhibitor treatments in cancer, if you don't have the certain microbes, these drugs are not working.
Interesting.
And that's probably more to do with the immune system, though. That's probably more to do with immune tolerance and various other things. And so what they're doing, there's a study out last year where they took a fecal transplant and were able to change the microbiome and then be able to get the drug to be working.
Interesting. One of the things, you know, one of the common denominators based on all the things that we've touched on so far seems to be the diversity of microbiomes. So within an individual. So, you know, you talked about the differences between people like us and hunter-gatherers. You talked about old versus young. And it seems in almost every case, like a more diverse microbiome is better and a more homogeneous one is going to have...
With one exception. One big exception. In early life. So in early life, the ideal microbiome is the least diverse. Because... so we know this from studies where we compare vaginally born breastfed infants compared to C-section born bottle-fed infants, for example, at two extremes there, you know. And what we want in early life is we want a lot of the bacteria, the good bacteria that are just there. We don't want diversity. We just want things that are just going to be doing the good stuff. So we have these bifidogenic bacteria that are there to harvest the goodness out of breast milk, for example. And so that's an example where less diversity can be. But you're right. On the whole, as we age, then you want, like in all aspects of life, diversity is really important. But that's just an interesting exception in early life.
So, you know, on this subject that you just alluded to, when you think about an infant child and their ability to metabolize the natural food that they're going to consume, which is breast milk, how much of that is, it sounds like a lot of that is microbiome connected, and how much of the infant's microbiome is being inherited through the birth process itself?
Oh, this is a great question. So two parts there. Let me deal with the breast milk first, because it was really surprising to me many years ago when I learned that human breast milk is among the most complex of all types of milk that there can be in the mammalian world. About 20 times more complex than that of bovine milk, which we use in formulas. And so evolution doesn't give us complexity without some reasons, usually. But what was really surprising was that these sugars, that these complex sugars, these human milk oligosaccharides that I'm really alluding to when I talk about complexity, that these sugars cannot be broken down we have no capacity to break them down in the body. So why would evolution do that? And the reason that evolution is doing that is because the microbes were there first, and the microbes are doing that job. And so microbes take these human milk oligosaccharides and break them down into key chemicals like sialic acid. Sialic acid is a really important part of brain development. So that is really intriguing data. And it may, and I put all caveats out there, but it may reflect why breastfed infants in large-scale population studies show a little higher IQ than bottle-fed infants overall, population-wide. It also is in line with work that Jeff Gordon's group have been doing in sub-Saharan Africa, where they've been looking at in moms that are not able to produce this milk, that there is a less of the sialic acid being formed in the infants, and then there's stunted growth and cognitive decline, and all of that being driven through this lack of a prebiotic, which is essentially what these human milk oligosaccharides are. And they've done elegant work over the years, both in the human population, but also in mouse models and pig models, to really show this interrelationship between the microbiome and the diet in driving brain developmental changes. And it offers, huge ramifications for our global public health policies in terms of how we manage that. So it is quite, we work a lot with industry in Ireland. Our funding model for research is very much an applied aspect. So we often work with industry, and often these industries are trying to recapitulate human breast milk in some way. And it's just such a hard job, because nature has done it so well for what it's doing. And we have relatively low breastfeeding rates in Ireland. So it's one of these things we really need to get across. Regarding the other part of your question, which was about vertical transmission, or I think this is a really important question, and we now we know a lot more about this. I like to refer to, so basically during pregnancy, the mom's microbiome changes. It adapts to the needs of the mom metabolically and every other way, but it's really getting ready to be handed over at birth as a kind of a birthday present to the infant to basically start programming the infant's immune system. For the most part, the infant is taught to be sterile in utero and gets the microbes as it emerges through the birth canal. And when in that, then this is kind of a really good example of potential epigenetic modifications that can occur, because we get vertical transmission from one generation to the next. The activation of the immune system starts pretty rapidly, and therefore anything that upsets this, such as being born by C-section, which you basically have you kind of drop the baton in an evolutionary relay race, or antibiotics, not breastfeeding. All of these things will factor in relatively to change these early life microbes that will program the developing immune system and body overall. So we are, one of that's one of the other things from a public health perspective. There are certain countries where C-section rates have just gone skyrocketing. Brazil is, parts of Brazil is 70%, China, but even in Ireland, it's doubled in 30 years. Parts of the US, it's gone really, really high. And so the question is what are the consequences of that? And so as I mentioned earlier, we worked on our animal model of this and show that there are enduring effects. But we just published a paper, which is another one of these papers that, take it with all of the health warnings. It's a small study, but it shouldn't have worked, but it did. And we got funding for it, where we took 40, I was going to say normal, but Irish medical students, and stratified them based on their mode of delivery. So we put 40 C-section and 40 vaginally born, and we bring them into the lab, and we have really good ethical ways of stressing people. And so we found that those born by C-section respond to acute stress from an immunological and behavioral point of view much greater. We also then wanted to look at chronic stress, and this is before the pandemic. So we wanted to see, how could we, if people were born by C-section, would they have a different response to chronic stress? But we had to do this in an ethical way. And the best way we had as educators is submit our students to exams. And so we can chart a variety of biomarkers over the examination period. And we found, for example, like the Cohen's perceived stress scale, which is a very well-validated marker of stress, was really higher over the exam period in those born by C-section versus not. Now, again, this is many other things could be at play here. We don't know why they had to have C-section. This was, some time ago, but our animal data backs it up very nicely and shows that the microbiome is what's playing, because in the animal studies, we could reverse these changes, these social deficits, by fecal transplants and by feeding with bifidobacteria. So these two questions you ask are really relevant to this perinatal critical windows idea. Because in neuroscience, we talk about critical windows a lot, from visual development to sensory and cognitive development. But there's probably also a critical window in our microbiome that we really need to watch, and how these two talk to each other is going to be really important.
So, I mean, based on everything you've said and just all of the interconnectedness here, have you ever thought about just how much, in modernity, we've essentially just completely removed ourselves from the ecological context that our species evolved in for so long? How does that make you feel in terms of your optimism or pessimism for our species? Because when you look at all of the chronic conditions, whether metabolic or psychiatric, and all, how much of this is just due to us, basically inventing our way out of the ecological context that all of this stuff was formed in?
It's a great question. And I do think about it in different ways. One is in the last 2 years, what we've just done. Just look at what we've done. At least here, we've had multiple lockdowns, social isolation, changed our whole social behavior. Then cleanliness and all of the effects we're having on our microbiome, even when we do. So we're affecting our microbiomes and we're affecting our social behavior. There will be many PhD theses in the future on this from a sociological and other ways. So I do reflect on it in that regard. Am I optimistic about us as a society? Yeah, I am, because I think, we are still a social species, and we want to talk, and people want to listen to you, Nick. And so I think, yes, we're doing things to damage our world, but we've been doing it, for centuries, and we'll continue to. And we'll continue to have wars, and we'll continue to have pandemics and various things. And we will prevail. We are living longer. We are living probably healthier. We're just understanding this more. But as long as we remember that the microbes were there first, and we need to look after them.
So one of the last things I want to ask you about is, given all of your experience and everything that you've learned, how has that affected how you look after your own microbiome?
Yeah, everyone asks me that. It does make me more aware of what I'm doing. I'm also, do I take probiotics? No. Do I? My lifestyle, well, it also changed a lot over the pandemic, but I used to be on a plane like 3 times a week, and, you're running around, and it's very hard to be looking after your gut if you're in an airport all the time. I can tell you that. So with the pandemic, it got me to reflect a lot more on my own health as well, and understanding what I need to appreciate with it. And with my kids, as my kids get older, I'm trying to foster in them. The one thing I do that people always want to know, what does he do? Like I could say, there are things that I can tell you are good for your microbes, but I don't necessarily do them. But the thing I do is I take fermented foods, which we have no culture of in this country, in Ireland at all. But I take milk kefir and try and have it every day. And the days and I haven't had it the last few days because I put it in.
What is it?
Milk kefir. Kefir?
Yeah, what is that?
Kefir. So this is a fermented grains that it's a ball of bacteria and yeast. And it's one of the 3 Ks. The kombucha, kimchi, and kefir are like the 3 fermented foods that people talk about overall. Why I mention fermented foods is they don't cost anything except what you put them in. So you can have water kefir or milk kefir. So if you buy milk, that's the only thing and you ferment it overnight, and you take it every day, and it's really good for your microbes. And we've done studies in animals where we've given kefir to some mice with autism, and they have better social behavior. Mice with autism-related symptoms, I have to be and we've shown that they have positive effects on stress. We've done a study in students where we've really ramped up their levels of kefir and fermented foods, and also their levels of fiber, and we've shown that their stress levels and their sleep quality gets much better. So, but the good thing about it is that it's a solution that we can offer to people that might benefit from it most. Not everyone wants solutions that are going to be in Whole Foods. Mm-hmm. So you need to come up with, for certain populations, you need to democratize. We need to democratize gut-brain axis health. And so that's why I also like to tell people about it. Overall.
What does it taste like?
It is like, so I got a trick on this. So my wife used to like it a lot, and I was like, oh God, so it tastes a bit like buttermilk or sour, it's a tangy flavor. But what I do is I ferment it overnight, and then I leave it in the fridge so you drink it cold, and then I put in some other yogurt, flavored yogurt in with it. Other people put honey or some, and then it tastes like the yogurt, just with a tang. It's a tangy, flavor on it. It's really good. It's really good.
I see. And when you say you ferment it overnight, like what exactly does that involve?
It means you take milk and you put it into the milk, and it's really, it ferments the milk.
I see. Okay. So you're buying something that you add to milk and basically just let it sit for a while.
Yeah. Yeah. And then you keep it again, like so it becomes your own. People have these like they do with kombucha. They have their own mothers. So you keep these kefir grains, and you put them into the next one of milk, and they grow, and then you give it to your friends, and then you pass it on, and then it becomes it travels with you like that copy of Ulysses you'll never read.
I see. And does this only work with like animal milk? Is there a particular
it works best with animal milk. I don't think it works so much with plant-based milks to the same level. But there are other fermented plant drinks and other fermented. I just think fermented foods. People think of them as sauerkraut and things that are not that flavorsome, and pickles and stuff. But they're a really good way of really giving bacteria back to your gut.
Interesting.
And I have data now in animals and humans, so I'm really, and we have a really nice team now working on this to do more comprehensive studies, and we've just got some funding from the US to look at this, and it's really exciting to in humans to see the temporal effects of it, what's it changing, and how is it changing in individuals versus otherwise. The other thing that in relation to practical aspects is in circadian rhythms and understanding the relationship between sleep and the microbes. And so my microbes, so it may not just be what you eat or what your microbes eat, but when your microbes eat. So my microbes are just so, they love carbs. They just do, and they're, but it's one of these things that personally that I think some of the beneficial effects of intermittent fasting, time restricted eating, etc., is now being seen to be driven by the microbiome, and we're really investigating this at the brain level, at the level of suprachiasmatic nucleus, in terms of the clock signaling, and trying to understand this because I think again everything being connected, but sleep and the microbiome is really relevant, and that's why I don't miss my jet lag days at all.
Interesting. Is there any, I'm sure you've got a bunch, but what's like one area right now that you guys are actively pursuing that you think is going to be really exciting just in terms of giving us some interesting results in the near future?
Okay, so we didn't talk. So I pretended to talk to you about the microbiome for this last hour and a half, but I actually only talked about the bacteriome. And so one of the things I'm really excited about right now is an area of the microbiome called driven by bacteriophages. Bacteriophages are viruses that target bacteria. So what if you have bacteria you don't want? And, one of the best ways is to get viruses to selectively kill them. And viruses have got a bad rep now with COVID, but like not all viruses are bad. And so this is a really exciting way. And that's something that we're working on right now in our animal models to show that maybe some of the effects of stress, could be alleviated through this type of mechanism. Really precision, interesting. It's going to change a little bit how we think about these things. So I'm excited about it.
Interesting. But the basic idea is if you've got certain bacteriophages that are adapted to infecting and killing specific species of bacteria, you could leverage that to alter an animal's microbiome in a beneficial way.
Yeah. Or you take them, like one of the experiments we're doing is we've taken the microbes before you get stressed or before you go through a negative effect, and you take the bacteriophages from there, and then you put them back in at the end, and make sure that they're able to work again and be able to counteract some of the effects.
Interesting. Well, we covered a lot of ground, John. Thank you for your time. And if you have any parting thoughts you want to leave people with, go for it.
Not really. I mean, it's been a real pleasure, Nick, and thanks for chatting with me. Overall, I guess the things I like to remind people is that unlike your genome, which not a lot you can do about except blame your parents and your grandparents, your microbiome is potentially modifiable, and that gives you great potential agency. But we have a long way to go. There's a lot of snake oil out there. Watch out for it. Look for evidence-based. Look for where there's data to support claims and data to support things. And if someone is telling you something, be careful. That's kind of where I'm leaving it.